Okay, good morning, everyone. Everyone in the room and everyone online, thank you very much for making time to come and attend this presentation. So I'm just going to give a quick introduction. Yeah, a pleasure to introduce Maisha to you. Maisha has been at SAIAB on a 7-month internship. She is registered at the Federal University of Rio de Janeiro in Brazil and currently doing her PhD. She came and spent a period of time with us for one of her research chapters. And her work is focusing on looking at how depth from shallow to mesophotic depth, so 100, 150 meters, so depth by geographic region and ocean basin influence the functional structure of fish assemblages with a specific focus on how things change as you move to the mesophotic region. So yeah, Maisha will be presenting on her chapter that's comparing the ocean basin scale questions today. And yeah, it's obviously been a really great privilege having Maisha here with us for the last 7 months. She's been working a lot with our MARIB team, helping in the field, helping students, and has become a really important member of our group. So we're going to be very sad to see her leave in a couple of weeks' time. But yeah, we've really appreciated her being here, and we look forward to hearing about her work. Yeah, thanks, Maisha.
Thank you, Ryan. Hi, everybody that is here. Hi, everybody online. Thank you so much for being here. Yeah, actually, following Ant's words, I'm very happy to be here, and I'm very happy to make part of this global network and global partnership collaborating with Brav Data. So I'll be presenting to you today what I've been doing here for these last 7 months. It's been a lot. So until now, it will probably change. The title of this work is Small-Scale Processes, Convergent Outcomes, Reef Fish Functional Vulnerability in the Mesophotic Zone. But when you talk about the Mesophotic Zone, I don't know if everyone is familiar to that. So let's start with the beginning. What is the Mesophotic Zone? So we call mesophotic everything that is between 30 and 150 meters depth. But we know that as the word says, mesophotic, it depends on light. And light penetration rates can vary depending on the region that we are. So if we are more in the tropical region, it can penetrate more through depth. If we are in temperate or regions with a lot of matter suspension and turbidity, it can penetrate less. And it can also vary depending on the season that we are. So we are still trying to define in the literature these boundaries of the mesophotic zone. But to make it easier for this work, we are working between 30 and 150 meters. And we can see huge differences between these photos, right? We can see in the first one, a shallow reef with a lot of light and a very well-known corals and fishes. And in the second photo, we see a darker place with different benthic composition and species that are more difficult to be seen in the shallow environments. So what changes from the shallow zone to the mesophotic zone? Of course, as we have a decrease in light penetration with depth, we have a decrease in temperature with depth, and we have also changes in nutrient availability and photosynthesis rates, and the biotic component of the environment, I mean the species, they respond to that changes. So how do they respond? We're still trying to figure out, because the mesophotic zone remained as a knowledge gap for many, many years, because it's too deep for us to be there so easily. So how do we get there? If deep diving is becoming more usual now, but it's not very safe. I mean, it's safe, but it's not for like everybody. I think the people doing that are kind of crazy. But yeah, we have the remote imagery that we are using a lot, and it's helping us a lot. We have ROVs, and we're still trying to disentangle the patterns and the processes underlying these patterns. But we have an agreement between researchers that we have a decrease in richness from many groups. So we have a decrease in reef fishes, we have a decrease in corals, and we have a turnover in the strategies that these animals rely on to use their resources. So as while in the shallow reefs, we have a more autotrophic strategies being performed, while we are going deeper, we have a turnover from more heterotrophic strategies. Even those corals that are in the shallow and in the mesophotic zone at the same time, they change their feeding strategies from autotrophy to heterotrophy, or at least mixotrophic strategies in the mesophotic zone. And we have a decrease in coral richness, algae, and an increase in sponges and more heterotrophic groups and species. And what about fish? That is my study object. So when you compare the shallow zone to the upper and lower mesophotic zones, we have a great turnover in species richness. It means that the species, they're being replaced with depth. And even between shallow and the upper mesophotic zone, that we can expect more connection, more ecological connection, we see great values of species turnover. And when we go further in depth, we see that this turnover only increases. And how it happens? I mean, who is where? So we see in that, if I do that, do you see the mouse?
Yes.
Oh, okay. So here we can see a comparison between the Atlantic Ocean and the Pacific and how families respond to depth. So reef fish families. We see a decrease in the majority of families. And we also see here, we see the comparison of trophic groups. So herbivorous families as the Acanthurigi family and the Scarini group, I mean the surgeon fishes and the parrot fishes, they rely on algae, detritus, corals, and as these groups, they reduce their richness and abundance with depth, these fishes also respond to these changes. But we have an increase in another families. So in the Atlantic, we have an increase in the Serranigi family. I think in English it's the basses, right?
Serranidae.
Yeah. Groupers, okay. And an increase in the Catodontidae family, it means the butterfly fishes. While in the Pacific, we see a decrease in the Catodontidae family. So it can change depending on the ocean and its evolutionary history and biogeographic context. But the most abundant trophic groups that we have with that in the mesophotic zone is the planktivorous fishes and also the mobile invertebrate fishes and carnivorous that can be in like a great portion of the depth range. So, of course, we have these species that can inhabit both shallow and mesophotic depths. But even these species that can be in the whole depth gradient that we are studying, they have their preferred depth range. So maybe we find 1 or 2 or maybe 10 individuals in one zone. But if we go deeper or shallower, we'll find more of them because they find more resources for their strategies and the functions that they perform in that preferred depth zone. So we also find a great endemism at depth, and we're still trying to understand why we have these huge endemism values and rates with great depth. One example is this beautiful fish. Its name is Tosanoides aphrodite. It's a Brazilian reef fish. It's being found in the St. Peter and St. Paul rocks. And until now, we think it's endemic to that island, but we're still we're not sure because we're still lacking a lot of research in the mesophotic zone. Maybe this fish it's in islands like in the surroundings, and we still don't know. But I'll take 5 minutes to take the story of this fish because it's very cool. So it was described in 2018 by one of my supervisors, and he was diving around 120 meters depth in that island. And when you dive so deep, you have just a little few minutes at the bottom because of the pressure and the gas changing. So they were very focused on the goal, that is collect this fish to describe it. So they were like, okay, quick, doing stuff. And the cameraman that was filming, he started to scream, shark, shark. And they were collecting the fish and they did like that. No, okay, don't bother me. And I don't know how to say it, sorry, but going up again from these deep divings can take hours and hours to decompress. So they took like 6 hours coming back, and they just discovered the shark when they were on the boat again, seeing the video. So the cameraman showed them the video, and they were like, "Oh my God, I didn't hear you, and I wasn't paying attention because I was so hypnotized for this small little fish. It's so small, so small." So they named this fish as Tosanoides Aphrodite, as the Greek goddess of love and beauty. Yeah, so we're still in these stages of the mesophotic science. We're still discovering who is there. So if we don't know who is there, how can we talk about patterns and processes and functions and how to protect these environments, as we don't know what's happening there? Okay, we have some data that can help us on that, but we still need so much more samples on the mesophotic reef and different type of methodologies being applied them. So BRAFs, deep diving, telemetry. I don't know how to install the receptors down there, but we must. ROV, yeah. So please, let's sample on the mesophotic zone. Okay, and until now I'm talking about who is where in the depth gradient, and it's very, how can I say it? Sorry, it's very related to environmental filtering, so temperature, food availability, but of course we have biogeographic processes happening also in the mesophotic zone that shapes the patterns that we see. So we found in 2023 through this article, this scientific paper, that the biogeographical processes shaping the shallow zone from higher richness in the coral triangle, from lower richness to its adjacencies, like in a longitudinal way, it doesn't happen when we go further in depth. So the upper and the lower mesophotic zones, they are more similar between them globally than the shallow zone. And I don't mean here they are similar in the species composition, but in the numbers of species. So of course, who is where, it depends on the evolutionary history of the ocean basin and some other evolutionary processes such as speciation and dispersal, but they are more similar in richness values, the total numbers. So we can see that we have a great decrease in species richness in the Pacific Ocean and a lower decrease in the species richness in the Atlantic, since in the Atlantic we have less species richness in the shallow zone. So of course this decline will be less pronounced. But okay, until now I'm talking about richness and who is where, but what about the functions being performed in the mesophotic zone? What are the ecosystem services that these environments give us? So we don't know much yet about it, and the research being done in the mesophotic zone, considering reef fish functional diversity, they are very concentrated in oceanic islands in the tropical region. So we know some patterns and processes happening there. We can find very similar values of functions being performed in the lower mesophotic zone than in the shallow zone. But of course, as we saw that the species richness decrease, these functions are being performed by less species, and it can cause vulnerability state in the mesophotic zone. But how does it work for the coastal region? And how does it work in a latitudinal gradient? We still don't know. We're trying to disentangle that. That's we are trying to do here in this chapter of my thesis. So my main objective is to compare the transition of reef fish functional diversity from shallow to mesophotic depths in a latitudinal and across shelf gradient. So we can see here our sampling design. We have samples in the tropical, subtropical, and temperate regions, and in the oceanic islands of the southwestern Atlantic and the Western Indian Ocean. It's been a great network and a great data collaboration that's allowing us to try to disentangle this pattern. This data has been collected since 2013 until 2024, so it's a lot of data. And to assess the functions being performed by fishes, we are using some functional traits related to the flux of matter and energy through environments and how fishes behave. So we are using diet, body size. The sociability means how they aggregate in large schools or small schools. Mobility, position in the water column, the period of activity, the geographic distribution of them, and the depth range that they inhabit. And I won't take too long in this slide, just to show you the analysis that we made, and it's a lot of analysis. So I know there's a lot of students that are starting the stats now, and it can be quite stressful. So as I'm going through that now, if you want help and if you need if you want some opinion or suggestions and help, I'm here to help. I'm glad to help. Okay, so after all of this stuff that we did, okay, we have our previous results. So here we can see the species richness per region, and you will see that every first box plot, where am I? Okay, every first box plot is the shallow zone, then the upper zone, and then the third is the lower zone in the Atlantic tropical region. We don't have the lower zone, unfortunately, yet. But yeah, so we see a decrease in species richness in all our regions sampled. It's totally, it's accordingly what we already knew, but we see a stronger decline in the tropical and subtropical regions of the Western Indian Ocean than in the other regions, of course, because these regions harbor higher species richness in the shallow zone. And as the lower zone is more homogeneous globally, we expected to see this decline. We can also see that the lower zones here, they're very similar, but we see that the temperate region and oceanic islands, they behave as a different world, like they have processes happening there guided by environmental factors or geographic factors, considering the oceanic islands, since such as isolation and their geography, they lead to these patterns that we're seeing here. And when we go to the functional richness, it means the amount of functions being performed in each region, we see that the patterns here doesn't follow, they don't follow the patterns that we saw before of species richness. So in some places we see the decrease in species richness, but at different magnitudes than we saw before. And in some other regions, we see a different pattern. Here, for example, in the subtropical region, we see in the Atlantic, we see a decrease of functional richness and then an increase again, and oceanic islands in the South Atlantic also present this different pattern. So why it's happening? What's happening there that are driving these different patterns? We'll see now. Here we have, oh, I'm sorry about that. Here we have the functional spaces of each depth zone for each region. And we can see that the shallow zone occupy the highest amount of the functional space, but we can see also that the lower mesophotic zone and also the upper, they are not merely contractions of the shallow functional space. They occupy different portions of all functions being performed. So functions there in the lower and upper mesophotic zone, they are different from those being performed in the shallow zone. And we can see that in the majority of places, we have higher similarity between the shallow and the upper zone than the shallow and the lower, or upper and lower. It just doesn't happen here in the southwestern Atlantic subtropical region, as we saw with the functional richness. And yeah, we will see why. So yeah, I won't take too long here because it's a lot of data, but what comes to our eyes is that. So how these functions, they change through the depth gradient. Here we see that we have different functions being performed at depth. So here we have the beta diversity analysis indicating to us that we have a great turnover between shallow and the mesophotic zones, but there's a lot of numbers and it can be a lot. So let's just give you the general patterns. So we found more differences along the depth gradient in the subtropical region of the Atlantic and in the tropical region of the Western Indian Ocean. So we have great differences in the regions that harbor higher species richness. And also great differences in oceanic islands, and lowest differences in the temperate region. So it means that the shallow zone in the temperate region and the lower mesophotic zone, they are performing more similar functions. And it was very interesting to see that the total value of the similarity between these depth zones, they were lower than the general, but also the turnover values, they had a lowest proportion in the total dissimilarity. So the nesteadness values, it means the loss of functions with depth, not the replacement of functions, but the loss of functions happen in a great proportion in the temperate region. So environmental filtering happening there with latitude and depth promote this similarity in the functions being performed. And we will see that now with our models, where we integrated our environmental and biogeographical variables to the functional indices. And in the first row, we have the functional richness. The second is the functional specialization, and the third is the functional redundancy. So what we see when we look at this amount of lines is that we don't have a global general pattern happening. We have context-dependent patterns that are being driven mostly by thermal differences along depth and habitat type. So environmental filtering is very important, shaping the functions being performed in each depth zone. Although we don't have a global general pattern, we have trends that are similar and converge even between regions in the same ocean and between regions in different oceans. So let's start with the oceanic islands. They are in yellow, okay? Everything in yellow are the oceanic islands. We see that we have a maintenance of the functions being performed in the shallow zone, in the lower mesophotic zone. So the function breadth is the same, but these functions are being performed by less species. As we saw, the species richness decrease. So we can see this decrease in the functional redundancy, and it leads to a state of functional vulnerability in the mesophotic zone at oceanic islands. We also see an increase in functional specialization. So it means that species in the lower mesophotic zone, they are, how can I say it? They're splitting more their resource use strategies to use all the resources there, and we can infer a great niche partitioning with that that's happening there. It also happens to the tropical and subtropical regions in the Atlantic. So the tropical is everything in red and subtropical in green. We see in the tropical region the same maintenance of functions being performed along depth, but by less species, so an increased functional vulnerability. And in the subtropical region, we see this different pattern, right, of decrease with depth, but then an increase again in functional richness and functional specialization. And it's being driven mostly by the habitat type that we have there. So we have in the shallow zone, a lot of rocky reefs, even the southernmost coral reef in the Atlantic. It's in the subtropical region. And while we go deeper until the mid depths, we find more sandy habitats. They are more or less complex. They're less complex. Yeah, sorry. And while we go further in depth, we find rhodolith beds. So these rhodolith beds, they are allowing this reemergence of functions that being performed there. Although they are not very complex, we know that they are major habitats for reef fish. So we have this similarity in the functional richness between shallow and the lower mesophotic zone because of the habitat type. And we have an increase in functional specialization from shallow to the lower mesophotic zone. But as the same amount of functions are being performed by less functions, we see a reduction in the functional redundancy, and it means an increase in the functional vulnerability. And okay, so here in all these regions, we have the clear patterns of functional vulnerability. But in the coastal Western Indian Ocean and in the temperate region of the Atlantic, things are happening in a different way. So we see a clear decrease in functional richness in all coastal regions here of the Western Indian Ocean. And also these just few functions being performed in the lower mesophotic zone, they are more similar to each other. And we have more generalist species in the lower mesophotic. It's being driven mostly by temperature filtering that constrains a lot the resources available in the lower mesophotic zone. So what has to be done in the lower mesophotic zone is just like similar things. And as we have less species performing just a little bit of functions there, and they are very similar to each other, we have an increase in functional redundancy. And when we have an increase in the functional redundancy, we can assume that this environment is stable and is more, how can I say, is more protected in terms of impact. If we remove a species, this function will still be performed by other species, right? Okay, but it's very context dependent, and it depends on the type of impact that we are talking about, and it also depends of what is being performed in these habitats. So looking at the species there and their functional traits, we see here, it's a lot, but follow me. Here in the first row, we see the trophic groups along depth in the Western Indian Ocean. We see a decrease of herbivorous fishes, an increase in carnivorous and mobile invertebrates, and a decrease in the other trophic groups. And when we look at the body size of the fishes here in the Western Indian Ocean, and also in the temperate region of the Atlantic, so this purple line here, we see an increase in the macrocarnivorous, and here we see an increase in the size of the fishes there, so the body size. So we have more large to medium to large fishes in deeper zones. So when we talk about fisheries, for example, we target these macrocarnivorous and mobile invertebrate fishes as our primary, the primary species that we want to eat. And we, of course, look for the biggest fishes, right? So if we talk about fisheries in the mesophotic zone and how fisheries impact the ecosystems, although we have great functional redundancy, if we remove a lot of species and a lot of individuals as fisheries do, we can put this apparent stable status of the lower mesophotic zone in a vulnerable status, depending on, of course, the impact that we are talking about. So we can see that different patterns and different processes underlying these patterns along different regions promote different, different processes underlying these patterns promote the same state of functional vulnerability in the mesophotic zone, even in two different ocean basins with two different evolutionary histories and ecological processes happening there. So yeah, we're still discussing all these results. I know it's a lot, and I hope soon we will have the paper to show you guys, but for now it's that. And I'd like to thank everybody, and I really like to thank all people in Marip group and Ant that is guiding me so much through this work, and all of you guys at SAIAB that are helping me. Without you guys, I wouldn't be here like at this stage. So thank you so much. Okay,
Yeah.
sorry.
Yeah, thanks very much, Maisha. That was a really interesting talk. Yeah, some great results. But I'll open up to the floor for questions. I don't know if anyone in the room has some questions that they would like to ask. Taryn, go for it.
The previous slide with the pickup sticks of all the different colors over there. It's super interesting with the tropical species in the Western Indian Ocean. You've got that really big increase in body size, and I'm assuming that those are the big 6 gill sharks. Are those the big ones coming out?
Could be, and also just the bigger snappers and groupers as well will be coming through.
But then you've got literally the complete opposite in the Southwest Atlantic. So it's just really interesting. Very, very different results for both of them, but the oceanic islands seem to be more or less quite stable, where you've got, you know, more or less the same size across the depth ranges. I was also just wondering the distribution of your sites, where you had like the oceanic islands and the temperate and tropical, subtropical. There was the oceanic islands over there in the southwestern Atlantic. Why is that a tropical island? Should it not be like a subtropical? The lowest yellow dot.
This one, okay. So this is the Trindade Island. Regarding latitude, is in a subtropical region, but considering the oceanographic conditions, it behaves as tropical. So we consider this coastal zone as subtropical because here in the subtropical region, we have an seasonal upwelling regime that makes the water really, really cold in summer. But although they're in the same latitude, it doesn't happen here. and the water here is very clear and hot. So yeah.
Amazing.
And considering the body sizes, yeah, so we maybe to be sure why it's happening, we must analyze the sample, where we have the samples. Maybe if they are in protected areas, it can impact the size of the fishes. And we don't have, unfortunately, we still don't have the lower samples in the tropical Atlantic. So we don't know what happened here in this part of the depth range. It can increase or not, but it's still missing information, and it can changes this pattern. We will resolve it in my postdoc research. I hope.
Is this mean body size or
Mean body size.
for the community?
Yeah, mean body size.
So then the higher diversity in the Western Indian Ocean will be mostly smaller species also, so that might drive that lower. And then there's most, I guess, bigger now, so it shows quite clearly. Thanks. Is there any other questions on the floor? I have a question. I don't know if there's anything online first that we can go through.
No.
Okay, so I was wondering about your functional traits. We know that fish, throughout their development, they change, they go through ontogenetic changes in habitat use and diet and all that type of stuff. What effect, so that obviously wasn't accounted for in this study because of size, you didn't have size data for everything. But what do you think that effect would be? So if you were able to incorporate ontogenetic changes in traits, so a juvenile of one species will be different to an adult in terms of its functional role of the same species. But how do you think that would influence your results if you have thought about that?
Yeah, so I think it will definitely influence our results. Maybe, I'm not sure, if BRAFs are the best sampling method to capture juveniles because of their sizes, and they can be very difficult to identify at the species level because sometimes they're very similar between each other, or they mimetize, right?
Yeah.
The word, yeah, other species. But if we have how to account for the juveniles in the assemblages, we will have probably a change in these results because they perform a lot of functional different functions in the environment. Maybe a juvenile, it's a cleaner, but when it's an adult, it's another thing, like it eats corals, I don't know. Maybe they hide in some caves, but when they are adults, they are more like, and of course it changes the flow of energy, matter, how species interact, but in the marine environment, everything is more difficult. I mean, it's difficult to be there, and it's difficult to account for these intra-specific changes. So I would really like to use here the real body size that I am seeing in the BRAFs. But we have some samples, especially in the Atlantic, that are from single BRAF, monobravs. So we don't have the total length of every individual. Yeah, but I think even using the real body size, it would maybe change some things because we see like if it's a juvenile, but it's not like those that really changes the colors and the shape, not shape, but yeah. We can see more small fishes in the shallow or in the
Yeah, exactly. I think for the depth generalists, it could be something that fish that occupy a broad depth range and that undergo those ontogenic changes in their diet and habitat use. They obviously they perform different functions depending on their size. It'll be interesting to integrate it, even if it is just for the subset of the data. Next question.
Yeah.
next topic for research.
Oh my God, we have so many topics considering the mesophotic zone, and we need to be there. Braving, ROVing, diving, telemetry, everything.
Specimens as well, eh?
Yeah, and doing the taxonomy. It's very interesting that the rates of new species being discovered by hours per hour of deep diving. I think that the rate is about 5 species per hour diving. It's so much. So
Yeah.
we still need to know who is there.
Yeah. Yeah. Yeah, we need to do more deep diving.
Oh my God, not me. I will stick with the brass.
Cool. Are there any other questions? Well, thank you very much, Maisha. Yeah, it's good to see the work coming together and looking forward to seeing how it develops further. And yeah, all the best for your next adventures. So, Maisha's starting it's your postdoc, eh?
I need to finish my
Finish your
PhD.
PhD, but then
But
the
yeah.
next is with the Smithsonian Institute doing the mesophotic stereobrobes work in the deep in the tropical Atlantic. So yeah, good luck for that.
Thank you so much.
And
Thank
thanks
you.
for joining us.
Thank you.